This procedure begins with fixation, methanol and proteinase K treatment of the embryos, followed by overnight incubation with probe 1, digoxygenin or DIG, and probe 2, fluorescein isothiocyanate or FITC. Embryos are incubated with DIG antibody. Following color reaction with DIG, alkaline phosphatase activity is inactivated, and embryos are incubated with FITC antibody. Color reaction is performed with FITC, and embryos are fixed.
Hi, I'm Delphine from the laboratory of Rick Finnell at the Center for Environmental and Genomic Medicine at Texas A&M University in Houston. Today we will show you a procedure for performing two-color in situ hybridization in the chick embryo. We use this procedure in the lab to analyze the effects of anti-epileptic drugs on gastrulation and neural genes. So let's get started.
To perform in situ hybridization on chick embryos, first open an egg by tapping the shell with forceps and removing pieces of the shell. Remove the thick albumin with forceps and tilt the yolk sac with coarse forceps so that the embryo faces upwards. Using fine scissors, cut a square of yolk sac around the embryo. Remove the embryo from the yolk with a spoon and place in a dish containing ice-cold Depsy PBS. Under a dissection microscope, carefully remove the membranes